Okay, in this video we're going to talk about how things get in and out of the cell, or how things move across the cell membrane. So diffusion is how things move from high concentration to low concentration. And it has to do with one of the laws of thermodynamics, the second law of thermodynamics. It says that the universe tends towards disorder or entropy. So you can see in our example here, dye molecules are moving across a semi-permeable membrane from high concentration on the left side to lower concentration on the right side. And they would continue to do that until they reached equilibrium. So diffusion is the movement from high concentration to lower concentration. So here's an example of simple diffusion. Things move from high concentration to low concentration. For instance, if you opened up a jar of perfume in one corner of the room, it would diffuse until it was evenly spread throughout the room. This is a form of passive transport, which means it doesn't require any energy for diffusion to work. So here we see the movement of water. The little molecules represent water. Water also moves by diffusion. We're going to talk more about water a little bit later on, but it's called osmosis. So facilitated diffusion is diffusion that uses protein channels. Some things can't go through the phospholipid bilayer, so they need a helper. In this case, they need a transport protein. The channel moves specific molecules across the membrane. So each channel protein is specific for a specific kind of molecule that needs to move in or out of the cell. Again, because it's diffusion, there's no energy required for this type of movement. It's going to go from high concentration to low concentration. The difference is, in simple diffusion, it goes through the phospholipid bilayer. It would be nonpolar small molecules. Polar large molecules need a channel protein. So facilitation means it moves with help. All right. Active transport is using energy to move things against the concentration gradient. In diffusion and facilitated diffusion, things went from high concentration to low concentration. We say they moved with the concentration gradient. In active transport, things are going to move from where they're in low concentration to where they're in high concentration. So if you think about a rock and a hill, diffusion is like the rock rolling from the top down to the bottom. Active transport is like pushing the rock back up the hill. So in this case, again, there's a carrier protein, but in this case, it's not simply a channel, but it changes shape as it moves the molecule from one side of the membrane to the other. And you can see that in the little diagram there on the slide. So an example of active transport is a protein pump. Again, things tend to want to diffuse from high to low, but in active transport, we would take things from when they're in low concentration to move their work to where they're in higher concentration. So there's a cost involved. In this case, the cost is energy in the form of ATP, which is the form of energy cells use to do work. And we'll talk more about ATP when we get to energy. All right. There are many models or many mechanisms in which active transport works. In the example here, we have antiport protein where it moves two different molecules in opposite direction. But again, in both cases, they're going from low concentration to high concentration. ATP is involved. In the second example, we have Symporter, where they're both going the same direction. So this is Symport. So passive transport, simple diffusion, nonpolar, hydrophobic, small molecules, lipids, high concentration, low concentration. Facilitated diffusion, polar, larger, hydrophilic molecules in a protein channel, again go from high to low. So those are both forms where no energy is used. Active transport, going against the concentration gradient, passive transport with the concentration gradient. this case, we're going from low to high concentration. Needs a protein pump. Requires energy. So here you can see simple diffusion going through the phospholipid bilayer, facilitated fusion using a protein channel, and active transport using a carrier protein, an ATP. So what about large molecules? How do we get large molecules in and out of the cell? So this involves vesicles and vacuoles, some of the structures we talked about when we talked about the cell itself. So one way to get things in is endocytosis. And I always remember enter and endo kind of start with the same letters. So phagocytosis, or cellular eating, is an example of this. When a white blood cell engulfs a bacteria or virally infected cell, that's a form of endocytosis. Pinocytosis is bringing fluid and dissolved substances into the cell. Cell drinking is kind of the literal translation of this. Exocytosis is exiting. So here you can see an example of different kinds of endocytosis. Phagocytosis creates a lysosome, or creates a vacuole which will fuse with the lysosome to digest what was brought in. Pinocytosis, more non-specific, it's fluid and dissolved solutes. Receptor mediated, you can see there's those little Y things are receptors that the molecule has to bind to, creates what's called a coated pit. And then you have the vesicle inside the cell.